Welcome everyone to Life and Breath Foundation, our educational webinar. My name is Sarcoidosis. I'm a board member here at Life and Breath. We're delighted to have you here with us today for our very first 12 o'clock noon webinar. Today's topic is Sarcoidosis Genetics 101. And before introducing our esteemed speaker, I would like to share a few words about Life and Breath Foundation. Our primary goals are to offer sarcoidosis community effective tools, educate and manage their condition, and maximize their quality of life. To provide open and nurturing environment for those affected with sarcoidosis, allowing them to share their experiences, to build greater awareness in general and within the medical community to help combat this chronic disease. Life and Breath is a volunteer-led organization. Money we receive through donations and sponsorships goes directly to supporting patients with sarcoidosis. Some of the different services we provide are obviously this monthly speaker series. We have a Life and Breath Scholar where we have resident physicians going through their process for sarcoidosis training, patient support from sarcoid experts, patient support through sponsorships and education, and we also have My Life and Breath Planner, which you can learn more about on our website. So everyone here, we did receive several pre-webinar questions and we're gonna do our best to address those. Also, Dr. Rivera mentioned that during the presentation, if anyone has questions, please submit those questions on the Q&A and we'll do our best to answer those during her talk. So we'll strive to get as many of those answered as possible. If we don't get to everything, we'll see if we can get to them after the webinar. Now, it is my pleasure to introduce Dr. Rivera, but before we do that, let me also mention that there is a disclaimer we have to go through. This webinar will not give medical advice. No doctor-patient relationship has been established talking with Dr. Rivera during this webinar. Our speaker doesn't know you, your history, your medical background, and therefore just can't give you any medical advice. Please speak with your own physician before acting on any information you hear on this webinar. Now, without further ado, let me introduce Dr. Natalia Rivera. She's a geneticist with a specialized focus on the genetics and genomics of sarcoidosis. She possesses extensive expertise in the study of complex human diseases. She was appointed as an assistant professor in 2018. Dr. Rivera has since dedicated her research to advancing the understanding of sarcoidosis genetics across various populations and identifying relevant biomarkers. She established a multi-ethnic sarcoidosis genomics consortium, which encompasses well-characterized population cohorts aimed at cataloging genetic variations in sarcoidosis on a global scale. Additionally, her research includes epigenetics of sarcoidosis. Dr. Rivera, the floor is yours.
Thank you very much for the kind introduction and to allow me to present my work as well as to give a webinar on this subject. So we'll start. Yes. So now let's start with, I will introduce you to genetics, first Genetics 101. If you can move to the next slide, please. So first we're going to start with DNA, and DNA is our molecule of life. So usually every DNA is packed, very tightly packed into each cell in our bodies, and it has 23 pairs of chromosomes, which contains all the genetic makeup information that we have, and it contains it's about 20,000 genes that coded for proteins, and there is extra additional information. So it's about, DNA is about 2 meters long, more or less, and it has 3 billion units, nucleotide units that we take into account. And each of these units have a specific function. And this is what we tend to study in complex diseases. So from the DNA, we start each gene. It has an expression in different cell types and it has function in different cell types. And this particular one, some genes, they interact with each other, some genes interact with protein. So it's different physiological and pathological functions depending on how we study DNA. Next slide, please. So if you look into each cell type, So every cell has a nucleus, and in each nucleus there is DNA information. So we have 22 pairs of what we call autosomal chromosomes, so 1 to 22. So 1 being the largest chromosome with many genes, and these are just the chromatids that we have, the chromosome itself, and each of these chromosomes will have different types of genes involved. And we have also the sex chromosomes, which is defines the biological sex between males and females. Next slide, please. So DNA variations, they are, because I mentioned to you, there's over 3 billion nucleotides. So these nucleotides, we call them single nucleotide polymorphisms. And these are the most abundant DNA variation in the human genome. So far, we know that there is about 85 million nucleotides polymorphisms in these 2 meters of DNA in every cell that has a nucleus. And the DNA SNPs occurs every change in DNA nucleotide occurs every 300 pairs. So every 300 polymorphisms, you will have a change. And this is normal. Some of them are rare, depending on the population-specific ethnicity or ancestry. And some of them are, a majority of them are on average exactly the same in the human genome. So we usually tend to count those nucleotide polymorphisms that are more than minor allele frequency more than 5%, which is common across all populations regardless of ancestry and ethnicities. In addition to single nucleotide polymorphisms, we also have other DNA variations, which are the insertions, deletions, frame shifts, copy number variations, but you will see this more present into cancer than in complex diseases. In complex diseases, you see a lot of nucleotide polymorphisms implicated in disease risk, while in cancer has a different in terms of mutations. Next slide, please. So I wanted to give you, before I go into the genetics of sarcoidosis, I wanted to give you an overview of what genetics is and how we study genetics. So for every genetics, or what we call genotype, so a genetic makeup or background, which is the genetic of an individual of any organism, and in other words, a biological fingerprint, we also have a phenotype, and we tend to have, to study genetics, we tend to have also a phenotype. So in this case, we have a phenotype. It could be eye color. So there are certain genes that are corresponding are corresponding to eye color. There is, in terms of obesity, there is certain genes associated with obesity, or slim, being slim or being obese, or body mass index. And there's certain genes also in terms of waist circumference ratio or waist hip ratio. So genes are actually characteristics of, they are markers of our phenotype, and they will dictate what phenotype, how the genes are expressed will dictate the phenotype that you see. So here, for example, I also say that you can have height. Height is one of the more heritable phenotypes. It's about 99% heritable, while complex diseases, we don't see this trend. So complex diseases usually are not heritable unless we're talking about monogenic diseases. So complex diseases, we call them multifactorial or polygenics. And I will go more into this as we go along. Next slide, please. So how do we study genetics or populations in terms of genetics? How do we study diseases actually? So what we do is we usually try to shape a cohort, build a cohort of, let's say that we want to study sarcoidosis. So I will try to get a cohort of sarcoidosis that are well-characterized individuals, and I want to compare with something that do not have sarcoidosis. And usually we tend to have a case where we take the controls, or healthy controls that do not have the disease, do not have phenotypes, or individuals that do not have similar phenotypes that can give us bias in sarcoidosis. So we try to characterize the cohort in a way that can give us the most possibility to find associations related to the disease risk. And in this way, we're looking at the single nucleotide polymorphisms that we have available on each individual with different type of technologies, and we try to compare those individuals across that cohort. So each individual, or each position in the DNA of each individual is compared between the cases and controls to understand which of those nucleotide polymorphisms are associated with disease. Next slide, please. So an overview, and this was back in 2008, 16 years ago, uh, genome-wide association studies, which is one of the main, has been one of the main source of finding genetic variants associated with different diseases, was established almost 20 years ago. And in this study, this is a really beautiful framework which allow us to study the DNA across the whole genome to survey different nucleotide polymorphisms that we want to see what is associated with disease. So usually in a genome-wide association study, we establish a cohort minimum. We have certain statistics, biostatistics, minimum 1,000 cases, and we try to compare 1,000 controls, or sometimes to increase, depending of the budget also, to increase the number of controls so we can actually increase the number of chances to find those signals associated with the disease. And we try to have the control as well characterized as possible within the same ethnicities or ancestry, so we don't get biased due to the different ancestries. And we try, depending the macro, the SNP array or the technology that we study, that depends basically on the funding. How much funding we have available will dictate how many samples we can include in the study. And then from once we do that, once we have molecular profile in terms of DNA, the individuals, we try to study those and compare those that are the cases and controls at each particular point. And we try to see, using different statistical methods and different frameworks, we try to see those signals associated with the disease. So if you look at the in the third picture, if you look at the fourth picture, this Manhattan plot, this is actually for each signal that you see 1, 2, 3. In the first signal, you see that those each one means chromosome 1, 2 means chromosome 2. So these particular bins are representing chromosomes, and in each bin, in each chromosome, each dot represents a nucleotide. So depending on how much financial resources you have, you can profile. Either you can use whole genome sequencing or whole exome sequencing, or you can use a SNP array to profile as many nucleotides as possible in order to find signals that are associated with disease. And then after we find a signal, so a genome-wide study will not tell you that thing. Is a utility, is a tool to actually find genomic loci, to find not just genetic variants, but also loci that might be possible associated with your disease. And this is what we want to do. So a specific variant, sometimes some of these variants will be between genes. So we don't know where is the gene, but if we go into the next layer, which is the transcriptome, how the gene is expressed, we can see regulatory variants in these regions that we call them non-coding regions. We see regulatory variants that are dictating how the gene should be expressed. So these are very important for us in terms of genetics to understand which loci are implicated, whether they are inside a gene structure, that means in an exon, in an intron, or between genes, because with that we can tell which loci are more likely to be captured for this disease. So in order to zoom in into this Manhattan plot, what we call them, these bins of chromosomes, we try to zoom in using fine mapping techniques, and there are different statistical methods. So it's a lot of bioinformatics in genetics that we actually apply to look at the different signals. And then the next step is replication, which is extremely important in genetic studies, because if I identify signals that are associated with a particular disease, I also want to be able, I need to make it reproducible, and I need to make it a true finding. And by making it a true finding, that means I have to replicate it in another cohort. If I have the resources and collaborations, I could make it in a cohort of a different ancestry, because what I want is genes that are associated with a disease, regardless of ethnicity or ancestry. Because these ones will be the ones that there will be biomarkers. This is the ones that those findings will be the ones that there will be potential drug targets that we can look into it. So we want to find those genes that are possible, that will be available for everyone, regardless of their ethnicity or ancestry. So replication and validation is extremely important in genetic studies. And there here where you see the genotypes is what I told you about, how we see the different, how they are observed between the cases and controls in terms of allele frequencies. So we keep comparing for each dot, for each nucleotide, we compare cases and controls. So sometimes we can have 7 million, genetic variants associated that being surveyed. Sometimes we can have 2 million. It all depends on how much. And if you have all the funding, you can have the whole genome. And then after we have found, we discover the signals, we replicated the signals, then we go into the next step, which is the biological validation. So there are different methods. If we have tissues, or if we have nowadays with the available next generation sequence technologies, we can look, if let's say that we find certain genes that are important associated with the sarcoidosis, let's say. So let's look at the transcriptome. How are these genes expressed in a particular cell type? How are these genes expressed in a particular tissue? So we try to replicate and validate this biologically using other, either in silico or tissue type. So in terms of mouse models, in vitro, in terms of cell types. So depending on what the disease is, we try to replicate this. Next slide, please. So I want to emphasize that, I want to emphasize the essential reasons for why we need patients to be more involved in molecular studies. And I think this has been a setback in many of the molecular studies. I started my postdoc here in Sweden in sarcoidosis 10 years ago. And one thing that I faced was the different type, this limited number of cohorts, the limited number of samples that we could study. During my PhD, I was involved in cardiovascular genetics of cardiovascular diseases, where we studied thousands of samples in consortia. Sarcoidosis didn't have that. So 10 years ago, we didn't have, we have basically 2-3 groups that they were working together, and then some groups that they were working alone. They were doing studies, but these findings was not able to be replicated in other cohorts. And this has been a big drawback in sarcoidosis research in terms of genetics, because if you don't find those signals that are associated with the disease, or those genes, then they may be causal genes for the disease, we cannot move forward. So I wanted to emphasize that it's very important that you as patients get engaged in these molecular studies, because we do need more patients, we need more engagement, and we need more funding. Because if the stakeholders do not see these large studies, we will not get funding, we will not be able to find these associations, we will not be able to move and compete forward like in the cardiovascular field or the cancer field, which are already now in the precision medicine with AI. We are, as sarcoidosis field, we are way behind, and I think we can do better. So I want to emphasize that why genetics is important in sarcoidosis research, and then why ancestry and diversity is important to be included in the sarcoidosis research, because as I mentioned, we want signals that has to be replicated across populations, because we want to find genetic markers, we want to find drug targets that are specific to the disease, regardless of what ancestry or ethnicity. So I think this is very important to emphasize. And also how you can get engaged and how you can contribute to help us to move the field forward. Next slide, please. So as I mentioned to you, genetics is very important, and we have seen it already with many different diseases. I mentioned to you cardiovascular diseases. We see it with metabolic diseases such as obesity. We see it with cancer that are moving forward. They are already way far away from us. But genetics, we know from those studies that genetics play a big role in either a big role in terms of, like height, which is 99% heritable. So we know the genetic variants associated with height, or a small role. Certain diseases are very specific and they need more samples. So to any extent, we know that genetics is a play in sarcoidosis. We know that DNA variation it plays a role either in rare or common diseases. We know for rare, it's one gene, one disease. Sarcoidosis, or like any cardiovascular or any complex diseases, there are many genes. So it's where we call this is polygenic. And for this, what we call this is a genetic susceptibility or genetic predisposition. And then we know also from other studies and other diseases that environmental exposures are big, and lifestyle behaviors are big factors in modifying disease risk. So we know that they can modify the genes, and how they can do so is through epigenetics. That means due to environmental exposures, there can be certain epigenetic marks that are changing, are driving the gene expression differently than it was supposed to be. So this is very important to understand. Does anybody have any questions before I move to the next?
I just one question. How can we get involved in a genetic study?
Okay, so that's coming on my turn. So it's basically for every. So I ask the patients for every time that they have a doctor's visit to ask their health provider whether they will be, if they know any cohort, ongoing cohort or new cohorts that are going to be established, to donate their blood, to donate. Sometimes they have saliva kits. So how they can get involved. So to ask the health provider if they know any researcher in the area that they are doing genetic studies or establishing cohorts for genetic studies or molecular studies in sarcoidosis. I think this is one way to go. So the next thing is why genetics is crucial in sarcoidosis research. So I have mentioned to you, and I mentioned these terms, sarcoidosis is, we know from our studies that sarcoidosis is a systemic immune-mediated disease, okay? But we don't know the cause. So we know that there are many genes that are related with the immune functions or immune-related functions are implicated in sarcoidosis. And now sarcoidosis can be a disease that can be only one organ or can be multiple organs. It can have many comorbidities, but the hallmark of the disease is these granular malformations in the affected organ. Sometimes it could be two organs involved. Sometimes it's just one organ throughout the development of the disease. So the heterogeneity of sarcoidosis is quite big. It's quite wide, the spectrum of the heterogeneity. And from what I see in the studies, I see sarcoidosis, the definition, as an umbrella of many different phenotypes. And each of these phenotypes have their own genetic architecture, because I will show you just an example of two different clinical phenotypes, how the genetic architecture changes. So, and I believe that as you know, some patients develop comorbidities, this genetic architecture is slightly different than those that they just have sarcoidosis by itself. So this is something that we need to study as we move forward. But first, we need to study the background of sarcoidosis. What are the genes and genetic variants associated with the disease first. So the pathological hallmark of the disease is granular malformation, or this imbalance of different cell types that are clustering together in the affected organ, which eventually can affect the organ function, the tissue function, and eventually it will make you sicker. So, and it's a complex disease. And when we mean complex in terms of genetics, that means that there's environmental factors implicated, lifestyle behaviors implicated. So I call this as moderators of the disease. Exposures make the susceptibility higher. And also it's multifactorial and polygenic. Multifactorial meaning that there are many different triggers, as well as polygenic. There is not just one gene, but many genes interacting with each other, dictating. And this genetic architecture will change with the phenotype under sarcoidosis umbrella. Next slide, please. So the roles of genes and the environment. So multifactorial, we have genes and environment interacting with each other. And we believe that this is the interplay that is driving the sarcoidosis susceptibility and the onset of the disease. Polygenic, we know in terms of the molecular, there are many genes. One particular that we have known for many years is this human leukocyte antigen genes, which is an immune, very strong immune gene. They are related, is implicated. So we know there is one big driver, but we also know that there are other genes, and these are interacting with each other, either at the protein level, at the gene expression level, or at the epigenetic level, or even when you talk about environment at the metabolic level. So there are many different layers that we can look. So as I said, sarcoidosis, from what we have seen in our studies, is an umbrella of many phenotypes. And each of these phenotypes will have common genes that are associated across all, but they also have a specific genes, which we call them endotypes. And these endotypes are very critical to actually characterize them. And this is where we need our clinical healthcare providers, because they see you every day, every time, and they have a bigger picture of the disease, and they can characterize. So I always believe a well characterized phenotype can give you the signals in genetics. If we have a very obscure phenotype, will not give the right signals. So, and this is very important. So that's why we work very closely with a lot of doctors that are sarcoidosis experts that have experience in sarcoidosis. And we try to disseminate also this knowledge to the younger doctors that are coming to learn, because it's an art to characterize sarcoidosis, I believe, because it's so heterogeneous. So, and I just wanted to show you that, you know, in terms of, we have one part, which is genetics, then the next one, epigenetics, where the environmental factors, the lifestyle, and other, it could be deep inside molecular, intermolecular epigenetics, or due to external triggers. And then we have the environment or lifestyle behaviors, where we have nutrition, the drugs, pathogens, exposure, etc. Next slide, please. Okay, so in a brief study that we did, this was back in 2016. We have a bigger study now going on, but the idea, if you can go back to the previous slide, so I want to show. Yeah, so the hypothesis of that study was we use a specific chip, which is called immunochip. And the hypothesis of that study was, is sarcoidosis an autoimmune disease or immune-mediated disease? That was our first hypothesis, because we knew that it was an HLA, and here in Sweden, we have a big sarcoidosis syndrome because we have a large number of cases of sarcoidosis, where HLA has always been part of the clinical diagnosis. So when patients come for suspected sarcoidosis, the first thing is to actually do a HLA typing on a particular gene. And the next thing was, what is the genetic overlap between sarcoidosis and inflammatory disease? And to do that, we use what is called this immunochip, which was customized already by that in 2016. It was already available to study autoimmune diseases. So we adopted that technology to study that. So we took, and it's smaller, it's only 200,000 nucleotide polymorphisms. So it's about 186 loci, genetic loci. You know, I told you at the beginning that the genome, we have 84 million SNPs. So it's only a tiny little portion. And also there are only 20,000 genes. So we study here 186. So 186 and the nucleotide polymorphisms on 186 equivalent to 200,000. And then in Sweden, we have what is called the Lofgren syndrome. And the Lofgrens, they are very specific. There are not many in the US. It's rare actually in the US, but the Lofgren syndromes are sarcoidosis patients that have an acute form of sarcoidosis, which resolves in 2 to 5 years. While the typical sarcoidosis, which we call them non-Lofgrens, it has a more pervasive form. It sometimes develops in pulmonary fibrosis. Patients develop comorbidities. So we have, you know, these are the typical sarcoidosis that we see across the globe. But the Lofgrens are very specific. Those are very, very specific, and they have a very specific genetic makeup. So we use the Swedish cohort as a discovery cohort. We had about 3,100 samples, individuals. And then we have replication cohorts. At that time was Germany, very small Lofgrens, and we have the typical sarcoidosis 400, and in the Dutch 150 and 80, and we also use the Czech Republic. So these have been these four: Sweden, Germany, Netherlands, and Czech Republic back then were here in Europe the most available with genetic data that we had, and we decided to join forces and do an immunochip. Now, next slide, please. So from that study, what we had, so, and this is we call this Manhattan plot because I showed you before that each bean is a chromosome and each dot is a nucleotide within the chromosome region, and we try to compare those, right? So this Manhattan plot is an overview of genetic variants across the genome. So here we have about 200,000 variants in Lofgrens and non-Lofgrens, so which I call them sarcoidosis, versus the healthy controls. And we found that the genetic architecture is different. One thing on chromosome 6 is where you see this HLA region, which is this HLA gene. But if we take other, because we only have about 3,000, we don't get other signals. And we usually use a threshold of 510 to the -8 to capture those signals. So here you see a big peak on chromosome 6 on the Lofgrens, but a slightly smaller peak, half of the N chromosome in non-Lofgrens. Okay, so go to the next slide, please. And then when we try to condition for this particular HLA, which is the HLA DRB1, we try to conditions, we see on the left side, on the Lofgrens, that we see that the signals are shifting because we know that carriers of HLA DRB1 or 3, the middle one on the left side, so there's 1, 2, the second on the left, is actually the ones that we know that they're going to resolve within 2 years. So if those patients have an HLA DRB103, they have a resolving disease. The ones that they have HLA DRB115, they are more doubtful because we know from another study that HLA DRB115 is a chronic, it is an indicator of chronic development or chronic disease. In the non-Lofgrens, you see a much lesser. You still see, but you only see other on HLA DRB115, you see other signals coming, while in the other ones there's not. So we know that HLA DRB103 is a major interactor gene, is interacting with many genes across the chromosome. We know that from other diseases, but with this, we know that it also does it with sarcoidosis. So from this study we concluded that sarcoidosis is an immune-mediated disease. We cannot say that it's an autoimmune because we need more immunological studies, and we need to see whether they are autoantigens and all these things that you see, and there are certain characteristics for an autoimmune disease, and we haven't arrived to that conclusion yet. But I will show you more data as we move. Next slide, please. So I want to, because I saw some of the questions, I was able to see some of the questions. What is this HLA? HLA is a region. Okay, and then within this region, there are numbers of genes. Actually, the HLA region is inside what we call it the major histocompatibility complex. And we usually use this region for organ transplants or marrow transplants. But this region is the most important region in the human genome or any mammal organism, because this region dictates your physiology of immunity. And we have in this region, there is about 421 loci, genomic loci, out of which 252, 60% are classified as expressed gene. And then the other ones are, transcripts that we call them pseudogenes, and there are other ones that they are not determined as gene, but we haven't learned that function yet. But in most complex diseases, genetic studies of complex diseases, like in cardiovascular and other diseases, we tend to ignore this because this region is the most difficult for geneticists to study because it's very complex. They are polymorphic. Some of these genes are polymorphic. That means there is not just two nucleotides but actually it can be many nucleotides. Like the HLA-DRB1 has 15 different alleles. So it's very difficult to study, but it gives a lot of information. So we know that there is, within these HLA regions, there is 3 different sub-regions, which we call it MHC1, MHC2, and MHC3. And in sarcoidosis, we see that the MHC class 2 are actually the most associated with the disease, whether it's Lofgren's and non-Lofgren's. And there is a shared overlap between Lofgren's and non-Lofgren's. But in the Lofgren's, you see the whole MHC mapping, while the non-Lofgren's is only very specific in class 2. So from there, we concluded that the 2 phenotypes of sarcoidosis, these 2 clinical phenotypes, have different genetic architectures, and therefore have different manifestations, different phenotype architectures. So next slide, please. So here, I saw some of the questions actually regarding comorbidities in sarcoidosis. So, and as we know from epidemiological studies from big registries, the Swedish registries that we have here, which were established in 1968, we can study many different factors and relationships across phenotypes. In terms of genetics, sarcoidosis, as I mentioned, is behind. But what we see is that comorbidities in sarcoidosis, we see that they are autoimmune. There is kidney disease, cardiovascular disease, metabolic, liver. Sarcoidosis is an umbrella of many phenotypes of its own, but it also gets affected by many other comorbidities, whether they are drug-induced, whether they are due to environmental factors, or whether they are due to other factors, we don't know. But now, as I mentioned, the most important thing for us first is to catalog the genetic variation of sarcoidosis. And using that, we can actually use the genetic data already available for other diseases. We have over 6,000 different phenotypes of genetic data, summary statistics of genetic data available on other diseases. So we can use that information to see how those genes are related or correlated with sarcoidosis. So here on the right side, I give you this is called a Sarcos plot. That means there are all the chromosomes, so from 1 to 22, the autosomal chromosomes. And we see here, this is a study on diabetes, type 2 diabetes, and the comorbidities of genes that are correlated, that share between other diseases. So we know that diabetes is shared with kidney diseases, with cardiovascular diseases, with autoimmune diseases, with liver disease. So there are many different factors that they are shared. So we want to find those shared genes, because those shared genes can help us to identify, repurpose the drugs that are already available for the other diseases that can be available for sarcoidosis patients. So this is very important to identify those shared, genetic regions, genomic regions that are shared with sarcoidosis that can give us an explanation why sarcoidosis patients have these comorbidities. I recently went to the American Thoracic Society and I went to the poster session, and I saw so many posters of young residents that they are showing sarcoidosis with cancer, sarcoidosis with liver disease, sarcoidosis with kidney disease. There were many pictures and faces of sarcoidosis. But me as a geneticist, I think, okay, it's not just the phenotype. There is something underneath it. And that underneath it are those shared genes. There is something in those shared genes that are making the patient susceptible to develop a comorbidity. And what are those genes? This is the important question that we want to answer. So we hypothesize that sarcoidosis have shared genetic overlap, shared genetic architecture with other complex diseases that may be able to explain why sarcoidosis patients develop these other additional phenotypes. Next slide, please. So I saw a question regarding autoimmunity. And this is a slide on the right side. It's a slide of the patterns of genetic overlap across autoimmune diseases. And this also actually was used also to kind of develop the immunochip, because they look at the shared genomic regions, genes across diseases, and they've developed a chip that we can use. And we see that there are many genes, not just a lot of people focus on the HLA, but there are other genes apart from the HLA that are implicated in autoimmunity and immune-mediated diseases. And this is what we also see in sarcoidosis. The problem that we have is that we haven't had the number of samples in order to capture the other genes across the genome, because the HLA is so strong that that signal is going to overshadow the other genes. So and this is why I try to emphasize patients to please join, these cohorts, because these molecular cohorts will help us to get these big numbers of samples that we can actually capture those genetic signals that we want. Because if you see here, you see ulcerative colitis, type 1 diabetes, multiple sclerosis, lupus, rheumatoid arthritis, celiac, vitiligo, SPS, Sjogren syndrome. All these, they have a commonality, which are these shared genes. And these are not just on chromosome 6. They are across the genome. So, it's not just one gene, one disease, or one genomic locus on one disease. There are many locus. But for that, we need more number of samples, bigger cohorts, bigger different diversity in order to capture this. And this is already 12 years ago. So I believe now that, if we redo this particular study and include sarcoidosis, we will be able to see some of this map. And on the left side, you see these epidemiological studies. So there are many, if you look in the literature, from 2017 and until now, and I think there are even more earlier. So we have, from the clinical aspect or the epidemiological aspect, we have seen the overlap between autoimmune diseases and sarcoidosis. So they have seen it not just in one ancestry. So the first study is in Taiwan. We see them in many different. So regardless of ethnicity or ancestry, there is a shared genetic overlap between sarcoidosis and autoimmunity, for sure. So we see that, is sarcoidosis autoimmune disease? We don't know yet, but, we are on the way to actually doing different studies to look. So they have been these associations, and then the last one is from here in Sweden, is using it's a recent, it was just published on June 8th from colleagues here in the clinical epidemiology, where they show that there's familial aggregation using the Swedish population register. They found this connection in a case control family study where they see this. So, and I also here in Taiwan, they also see a similar case. So we know that there is a connection with this, but which genes are connected, we don't know, because we haven't been able to do this large genetic study first. So, and one advantage, if we find this, is that drugs are already available for these diseases. So it will be easier, once we know which genetics, which genes are associated with sarcoidosis, overlapping with other immune diseases, we can look at those drugs, and these drugs can be repurposed for specific sarcoidosis patients. And this will be much better than coming with a new drug where it's going in different phases, and you going on the clinical trial, where some patients will be respondents, some patients will not respond to the drug, and some patients will develop drug-induced comorbidities. So I think we can learn, from these big studies, and we can learn what is available on these diseases to actually move forward the sarcoidosis field. Next slide, please. So the next slide is regards the consortium. So when I started, I mentioned to you 10 years ago, were very small cohorts, and during my PhD, I was working with large cohorts, large consortia in cardiovascular. So So I said, why don't we just unite forces and contact more people and try to establish cohorts with what we have now and the knowledge that I have acquired while I was studying doctoral studies to make it a bigger and make a consortium. So it took me 10 years, but we are getting there. And my goal is to expand and to do the genetic variation of sarcoidosis across the globe. And right now we're looking into lung, but we would like to also look in ocular sarcoidosis, skin sarcoidosis, and cardiac sarcoidosis, which are the main ones and the big ones that we can get numbers in terms of patients. And the idea is to do genome-wide association studies across these populations, try to find those genes that are associated across all those populations, and then do, the file mapping, the functional in silico studies, and then the biological experimentation. Next slide, please. So I saw some questions about precision medicine. So this is a slide from cancer. So in cancer, because the available funding that they have had, they have been able to move much faster and much forward, fast speed. And the ideas of personalized medicine is that using your molecular information, the molecular phenotypes, so the genome, the transcriptome, the epigenome, the microbiome, the metabolome, we can be able to identify targets, either biomarkers for biomarkers for disease biomarkers, either for diagnosis, prognosis, treatment, monitoring, treatment, different types of biomarkers, or we can find those that are one treatment specific, responders, not responders, and also the clinical one, diagnosis, prognosis. So there are different ways to do, and I think this is a beauty to start this now and to engage as many people and as many researchers as possible to work in collaborative efforts, because we know as we put the different layers, more information we get. And we gain more information with these diseases, with sarcoidosis. Next slide, please. So, so precision medicine, and I mentioned to you, so it's before it was one drug made for derived from the small cohort and given to all the patients regardless of it. And this usually has been done from the smaller cohorts to apply to a massive population. Now what we want is to have larger cohorts to be more specific, and given your molecular makeup, they could be drug target specific. So I mentioned to you in terms of autoimmunity drug. So let's say we see a patient that have a cluster of autoimmune disease, then we can look at those autoimmune drugs that may be specific for that. There's a specific genetic makeup for that particular patient, rather than a patient that has sarcoidosis and metabolic phenotypes. So cardiovascular disease, then we look at the cardiovascular and see whether those drugs available in cardiovascular can be repurposed in sarcoidosis. So that's the idea. And this is how I think we're going to move forward. I think we are getting towards the end. I think I have one or two more slides. So how you can be involved. So adhering to donate biological samples, I think is very important. Either, depending of whether your physician has a lab or whether it has a collaborator. So either donating blood or giving a saliva kit for molecular studies, participating also in questionnaires and surveys, which are very important to characterize the heterogeneity of sarcoidosis, as well as participating in patient groups like this one, the Life and Breath, because it's very important to make your voice heard. Because if you make your voice heard, we can get more funding. And this has been a big limitation in sarcoidosis. We can do wonders, but if we don't have funding, we cannot do more. So it's very limited studies, very small studies, little scale. So we cannot make these big novel discoveries. So I urge you to get involved with the foundation and so we can move forward. So with that, I think this is my last slide. So we can help us together. We can do many discoveries, and we can have robust data, and we can have better solutions for sarcoidosis. So I think it's the end of my presentation. So thank you very much.
Thank you, Dr. Rivera. Dr. Rivera, would you have time for a few questions that are
Yes,
that have
yes,
popped up?
I
So let me
yes.
get some of them answered that are live. There's a lot of interest in getting involved with studies. How do I find who is studying genetics, doing research in genetics for sarcoidosis?
So the MESARGEN Consortium has, we are working on a website that we are putting the different cohorts and the different collaborators, and I'm also including the ones from the US. So I will include the names of those collaborators in the different, and then you can also Google those collaborators and then see how they are involved. And I think that's one also the MESARGEN website. I will, once we publish the results of the big study that we're doing with 15 different populations. We will make that data, those visuals available for people as well as stakeholders and other scientists see which genes are involved. So we're trying to disseminate as much information. So I will make, through the website, I will make available of the groups that they are doing genetic studies in the US.
Thank you very much. On that same line, have you collaborated with Johns Hopkins Sarcoidosis Clinic for past or future research studies?
Yes, we have established a collaboration with Dr. Michelle Sharp, and also Dr. Drake at the University of Maryland. So we are working on this. And also I am establishing collaborations I have collaborations already with Dr. Courtney Montgomery in Oklahoma. I'm establishing collaborations with other different geneticists in different states. So we try to expand this because I think it's very important to catalog this genetic variation of sarcoidosis and actually also get this heterogeneity in terms of the characterization of the disease.
Okay, similar question. How do I volunteer for any or all experiments or studies? I will do anything to benefit the field.
Yes, thank you. So once I put those collaborators on the website, they usually, those labs, they have a inquiry or a website that you can contact them through those labs. Every lab has the specific ones. We are not a coordinated center. I coordinate the molecular studies, but I can ask my collaborators to send me those websites that I can put them in the MESARGEN website, and then patients can move forward. Because I think right now we don't have, and I'm also working with other different in the European sarcoidosis associations that they can put this data available, because we also have this in the European countries where patients want to be involved, but they don't know where. So we will try to disseminate this information so they can reach out to the different labs that they are doing genetic studies.
Okay, just one or two questions. Are you able to share how sarcoidosis and MS mirror one another?
So MS is an autoimmune disease. So we know the genetic variants and genetic architecture of MS. I mentioned to you that they are sarcoidosis. It's an immune-mediated disease. I cannot say it's an autoimmune because we need more evidence and more data on this. So there are immune genes associated with this. So my suspicion is that they are shared immune genomic loci associated with autoimmunity or immune functions within these two diseases. And one way to do it is to, once we have, through the MESARGEN Consortium, we have done the big GWAS, we can use that to correlate with the genetic architecture of MS and other autoimmune diseases, and we can see those specific genomic loci. So that will give you some clues. It will not give you the whole picture yet because we are at the baby step. Sarcoidosis, remember, is much behind than the bigger fields that they are already in the precision medicine era.
Right. How can genetic testing help in clinical care of sarcoidosis, as most HLA genes are associative with certain features?
So how can I help? So I mentioned to you, we're trying to look for biomarkers, and these biomarkers can be either at the diagnosis, but most importantly is at the treatment monitoring and the prognosis. Because once you are diagnosed with sarcoidosis, we don't know which course the disease is going to take. And this is very important because we could be just simple adjusting your lifestyle behavior better, eating better, physical activity. We know from the autoimmune diseases that this makes it better. No, no smoking. This makes it better for the autoimmunity. So they might be similar, already established, but we don't know yet now because we don't have the big data, a big study for sarcoidosis yet. And we are working on this through MESARGEN. Yes.
Okay, so I think we're done time-wise. We run up against the clock. I'd like to thank you, Dr. Rivera, for a wonderful presentation. This is the first time we've had someone speak to the group on genetics and sarcoidosis. So thank everyone for attending. Thank you, Dr. Rivera, for a very interesting talk. I think people have a lot of questions. We'll do our best to get the answers to those questions online. And again, thank you very much. We do these webinars every month. So if you're interested in participating in a future webinar, just look at our website and register. We'll see you then. Dr. Rivera, again, thank you very much for the time you spent and the education you gave us and our patient population. Thank you very much.
Thank you very much. Thank you. Bye-bye.